This lecture covers Chapter 20, the subject of which is antimicrobial compounds. Antimicrobial compounds are grouped together in what are called chemotherapeutic agents. And we don't mean chemotherapy in the cancer-fighting sense of the word. Chemotherapy meaning a chemical agent that is used to treat a microbial infection. And the search for these chemotherapeutic agents was called a search for a magic bullet by one of the people who sought them out, Paul Ehrlich, because they were trying to convey the idea that they were trying to find something that would magically injure the pathogen while preserving the health of the host. And strangely enough, the magic bullet that everybody was looking for was the magic bullet to cure the sexually transmitted infection syphilis, which you might be surprised to know has plagued humanity for hundreds and hundreds of years. It's thought to have become a problem at the end of the 15th century when French troops invaded Italy and brought syphilis with them and caused 5 million deaths. So this is something that was far more virulent when it came into the human population and has slowly become less and less virulent over time. And that's a typical thing when things enter the human population. They're oftentimes really virulent, and then over time they become less so. So in 1495, you have the movement of syphilis from France to Italy, and by 1539, there are 1 million cases of syphilis throughout Europe. We fast forward to 1917, the First World War. There were 383,000 cases in the U.S. Army, and the Army brought that disease back to the United States. And in 1932, a notorious study called the Tuskegee Study of Untreated Syphilis in the Negro Male was begun. And the reason I say this study was infamous is it very unethically, over a period of 40 years, tracked known cases of syphilis in the population of Black people around Tuskegee, Alabama, and chose not to treat them in order to determine what would happen if a person had syphilis for an extended period of time. Now, that obviously is not the way that any research should be done, and it's been widely criticized and was used actually as a way of creating new protections for people within medical studies. And historically, syphilis was referred to as venereal disease. That became sort of a catch-all for sexually transmitted infection, but venereal disease specifically refers to infection with syphilis, which had to do with the name of the goddess of love, Venus, so venereal coming from Venus. And it used to be said that a night with Venus would get you a lifetime with Mercury, which was, incidentally, the first treatment for syphilis. So treatment with Mercury, which is a poison, was the only known treatment for syphilis until in 1909 in the laboratory of Paul Ehrlich, whose name might sound familiar to you from our discussion in Chapter 18. A breakthrough was made with compound 606, named so because it was actually the 606th compound that was tested for its efficacy against syphilis. And this compound 606, salvarsan, which was a synthetic arsenic, became the next treatment for syphilis. This arsenic caused far more problems for the syphilis bacterium than it did for the host. It was considered a magic bullet, though as arsenic, it is deadly if it's overconsumed, and it actually was really difficult to administer. It had to be injected, and when it was injected, it had to be dissolved in several hundred milliliters of distilled sterile water with minimal exposure to the air. And so it was a difficult treatment to have available for people in order to block this syphilis infection. Thankfully, though, there wasn't much longer to wait before another magic bullet was discovered by Alexander Fleming by accident in 1928 again found to be a magic bullet against syphilis. So in 1928, Alexander Fleming came into the lab and noticed that there was a fungus contaminating his petri dishes, and around that fungus he noticed that the bacteria were inhibited from growing, which may sound familiar to you because we have learned about zones of inhibition, and this is where we get that concept. He noticed that the mold Penicillium notatum was producing some kind of chemical which was excreted into the media and inhibiting the growth of those bacteria. It took about 12 years for that chemical, which was called penicillin as a nod to Penicillium notatum, to be clinically isolated and the effectivity to be determined. But when it was, as you can see from this great poster here, this disease, which is as old as creation, syphilis, is now curable by a much more specific magic bullet, the antibiotic. Now you might be wondering at this point, what's the difference between a drug and an antibiotic? Why did I refer to arsenic as a drug and penicillin as an antibiotic? And the difference lies in whether or not that compound is made by microorganisms or is synthesized in a lab. And so drugs are all synthetic. They have synthetic roots. Antibiotics, though they can be synthesized in the lab, are naturally produced by microorganisms. And you can imagine, if you think about what's happening on this petri dish, this fungal colony is excreting a chemical which kills off all of the bacteria, and that means that all of the nutrients in the medium surrounding that colony are available to him and him alone. So antibiotics have been around as long as microorganisms have been around, and they've been one of the ways that microorganisms fight with one another and ensure that all the good stuff that's necessary for survival is there for them, because they've removed all the competition. Antimicrobials come in multiple different kinds. Some of them are called broad spectrum, some of them are called narrow spectrum. If you look at this table from your textbook, the smaller the arrow is, the more narrow spectrum the antimicrobial is. So for instance, this streptomycin that you can see is useful against mycobacteria and gram-negative bacteria. So that's a fairly broad spectrum antibiotic. If you compare that to penicillin G, which has effectivity only against gram-positive bacteria, it's far more broad spectrum than that. But if you compare it to tetracycline, you'll see that tetracycline is effective against mycobacteria, gram-negative bacteria, gram-positive bacteria, and even some of the chlamydia and rickettsia. So capable of effective use against mycobacteria, which have that mycolic acid in their cell wall, against gram-negatives, gram-positives, and also chlamydias and rickettsias, which are wall-less intracellular parasites. So those would be antimicrobials pointing at prokaryotes. There are also antimicrobials against eukaryotic pathogens, fungi, protozoans, helminths, and then also against viruses. So again, broad spectrum, meaning you can use this against multiple kinds of microbes, and narrow spectrum, meaning you use it only with a very small number of microbes. And it doesn't mean, for instance, that penicillin G is not useful. It might be incredibly useful against those gram-positives. It just means that you can't use it against other things. Now, you might remember from our lab activities that we looked at 8 different antibiotics and tested their effectivity against 4 different organisms. And then we ranked those antibiotics, and we talked about the fact that some of them are broad spectrum and some of them are narrow spectrum. And from these data here, you would say that ciprofloxacin, or Cipro, because it has the largest zone of inhibition, is the most broad spectrum of all of these antibiotics, while penicillin, which has no zone of inhibition, would be the most narrow spectrum. The way these antimicrobials work is to target a particular portion of the bacterial cell and inhibit that particular metabolic pathway or structure. And if we talk about antimicrobial targets, we have 5 major targets that antimicrobials, both antibiotics and drugs, can attack and damage or kill the microbial cell. So the first of those is cell wall synthesis. So there are multiple kinds of antibiotics which inhibit cell wall synthesis. There are multiple kinds of antibiotics which inhibit protein synthesis at the level of translation. There are multiple antimicrobials which inhibit nucleic acid replication, which would include both replication of DNA and replication of RNA. So both DNA replication and transcription fall under this larger umbrella of the inhibition of nucleic acid replication. The fourth antimicrobial target is the plasma membrane. So there are antimicrobials which injure the plasma membrane and cause cell lysis. And then finally, the inhibition of an essential metabolite synthesis. So find something that the cell absolutely needs to make in order to survive and inhibit that process by inhibiting an enzyme, and you will kill the cell. So again, 5 targets of antimicrobial agents. You don't need to know the particular antibiotics or drugs that are used for each of these targets, but you should be familiar with those 5 targets. And we're going to talk about each 5 of those now. The first that we're going to talk about is protein synthesis. Note here there are lots of different targets of that translation mechanism that can be hit. You can bind to the ribosome, you can alter the shape of the ribosome, you can inhibit peptide bond formation. Notice I'm being very vague here. That's intentional. You don't need to know the specifics beyond protein synthesis inhibition is one of the antimicrobial targets. The reason I left this in here is because these are commonly used antibiotics, and it might be interesting to you to know what the mechanism of action of an antibiotic that you may have taken has. So what we need to know is protein synthesis inhibition. Second target was actually number 5 in the previous slide, and that is metabolite inhibition. And most of the time, metabolite inhibition is done by the use of something called a growth factor analog. And growth factor analogs are analogous. They are similar to a growth factor or a substrate for an enzymatic pathway. And because they are analogous to, they compete with the substrate for the enzyme's active site, and they perform that competitive inhibition that you learn about in general biology. So if you can inhibit the synthesis of a metabolite by giving the bacterial cell something that looks like a substrate for that metabolite, then you will stop them from making the metabolite, and you will stop them from growing and perhaps kill the cells. Now, this can be selectively toxic so long as you are using something that confuses the microbial enzyme and does not have an effect on enzymes in the host. So one metabolite pathway that's a common target for growth factor analogs is the pathway that begins with para-aminobenzoic acid and goes through a multi-step process that ends with the production of folic acid, which is used to produce purines, pyrimidines, and amino acids. So if you block this pathway, you block the production of folic acid, which means that the bacterial cell cannot make nucleic acids or proteins. And obviously, when you can't make nucleic acids or proteins, you will die. So how is this selectively toxic? And that's an easy one. You and I, human beings, do not make folic acid. We have to consume folic acid in order to make purines, pyrimidines, and amino acids. So you don't have any of these enzymes in your cells, so there's no way that you can be confused by this. And notice there are 2 antimicrobial agents which inhibit this metabolic pathway. Sulfonamides or sulfa drugs inhibit the first step in this pathway. They stop para-aminobenzoic acid from being converted to dihydroteroic acid. And then trimethoprim inhibits the enzyme which converts dihydrofolic acid into folic acid. And that allows this to be a much more broad spectrum treatment because only certain bacteria perform those first 2 steps. Other bacteria consume dihydrofolic acid and convert it to folic acid. So if you can hit 2 parts of this pathway, you'll hit the guy who does both twice as hard, and you'll make sure that you hit both kinds of organisms. So we are going to spend some more time talking about this, which means I do want you to understand the details of this use of the growth factor analog to inhibit the synthesis of folic acid more than you needed to understand protein synthesis inhibition. So let's look a little bit at this process. So the normal metabolic pathway is begun by an enzyme called tyrosine synthetase grabbing para-aminobenzoic acid and converting it to that dihydroteroic acid that we talked about in the previous slide. And then that's used from that point on. When a person takes a sulfa drug in order to combat a microbial infection, notice the sulfa drug is a growth factor analog for PABA, para-aminobenzoic acid. So it has that benzene ring, it has the amino group at the end of it, and so it is recognized by tyrosine synthetase as a substrate. But of course, when a sulfa drug is incorporated into this pathway, folic acid synthesis is halted because that sulfa drug is not dihydroteroic acid, and it won't be recognized by the next enzyme. So this will block the synthesis of folic acid and kill the microorganisms, and again, will not affect the human host because we don't have tyrosine synthetase, we don't make folic acid. Now, this sulfa drug, which you'll hear lots of different names for, sulfa drugs, sulfonamide, sulfamethoxazole is one, that's typically used in conjunction with that second drug we talked about in the previous slide, trimethoprim, in order to block two parts of that pathway. And the reason that's done is because sulfamethoxazole and trimethoprim act synergistically together. Together they work better than the sum of their parts. So synergism just refers to the fact that sulfamethoxazole and trimethoprim pound for pound work much better than you would think they would looking at the way they work individually. And by the way, sulfamethoxazole and trimethoprim are one of the three antibiotics that you use to differentiate streptococcal bacteria in your SSE unknowns, and the trade name for them is SXT, sulfamethoxazole trimethoprim, the S and the X from sulfamethoxazole, and the T from trimethoprim. So sulfamethoxazole and trimethoprim are both growth factor analogs that are used to block the synthesis of the necessary metabolite folic acid. So that's two of the targets of antimicrobial agent. Target number 3 is the plasma membrane. You might remember from general biology discussions that one of the jobs of the cell membrane is to maintain the shape of a cell. And those antimicrobial agents which disrupt the plasma membrane actually alter the membrane structure and increase its permeability. And when they increase the permeability of the cell wall, water rushes in. And as you can see from the transmission electron micrograph here, the cell blows up. One example of an antibiotic which alters the membrane structure is polymyxin B. Now, you don't need to know that detail, but if you look at the diagram here, polymyxin B moves through the cell wall, binds the cell membrane, disrupts the membrane. As you can imagine, as soon as that happens, boom, the cell blows up. So protein synthesis inhibition, metabolite synthesis inhibition, disruption of the plasma membrane. And then number 4, the inhibition of nucleic acids, which again utilizes things called growth factor analogs, which in this case are oftentimes referred to as nucleoside analogs because they're not just a growth factor analog, they are an analog of a factor that is used to build nucleic acids, which are called nucleosides or nucleotides. What you're looking at here are two chemicals. One is called deoxyguanosine, and you can see from that ring structure, you have the deoxyribose sugar. Attached to that, you have the guanine nucleotide. The reason this is called a deoxyguanosine instead of guanine is because this has not yet had its triphosphate groups added to it. Now, compare the structure of deoxyguanosine, which is a growth factor, a nucleoside that is used to make nucleic acids, with the drug acyclovir. Acyclovir has a portion that is absolutely identical to the guanine molecule, but what it's lacking is that deoxyribose sugar. And you might be able to see where this is going already, because remember, DNA polymerase needs that OH group in order to add subsequent nucleotides. So acyclovir is a growth factor analog of guanine. It can be incorporated into a growing nucleotide on accident instead of guanine, and when it does that, it blocks the synthesis of the nucleic acid. This is selectively toxic because, of course, every cell needs to make DNA, is that your cellular enzyme is not confused by acyclovir. The cellular thymidine kinase, which is the enzyme that takes up this deoxyguanosine and adds the phosphate groups to it, doesn't ever pick up acyclovir. It's not confused by acyclovir. It will ignore acyclovir, so it will preferentially pick up the guanine, deoxyguanosine, add the phosphate groups to it, make it into a nucleotide. That nucleotide will be used to build DNA, and that cell will function just fine. In a cell that is infected by a virus, a lot of times that virus will bring with it a viral thymidine kinase, and that thymidine kinase is confused by acyclovir. It does not recognize the difference between deoxyguanosine and acyclovir, so it will grab the acyclovir, add the phosphate group to it, and create this false nucleotide which cannot be used to make DNA, and assembly of the DNA stops in the virally infected cell, and the virus is not able to replicate. So this is selectively toxic. It will only work in cells that have been infected by a virus that brings with it a thymidine kinase that is capable of being confused by the growth factor analog acyclovir. All right, so that gets us through 4 of the antimicrobial targets. We've got nucleic acid synthesis, protein synthesis, metabolite synthesis, and the cell membrane. Our 5th and final target for antimicrobial agents is the cell wall. And for that, we are going to spend some more time talking about something called a beta-lactam antibiotic. In order to talk about this 5th target, the cell wall, I'd like to show you a video, because I think this video will really give you an idea of how the cell wall is synthesized and how different kinds of antibiotics work to inhibit the growth of microorganisms by inhibiting the cell wall.
In 1928, Alexander Fleming discovered that a fungus contaminating his petri dishes had released a powerful antibiotic that killed the surrounding bacteria. He named this antibiotic penicillin. Nearly 2 decades later, after years of intense cooperation between scientists, manufacturers, and government agencies, penicillin was made broadly available. In 1945, using X-ray crystallography, Dorothy Hodgkin determined the chemical structure of penicillin, a discovery that fueled the quest to synthesize the drug. In 1952, a form of penicillin suitable for oral use, called penicillin V, was developed. And in 1957, penicillin V was produced synthetically for the first time, laying the foundation for the development and synthesis of new penicillin antibiotics. Penicillin is effective for treating bacterial infections because it disrupts an essential process in the bacterial life cycle, the creation of the bacterial cell wall. The cell wall in most bacteria consists of a lipid bilayer membrane and a mesh-like peptidoglycan layer. Gram-positive bacteria build a thick peptidoglycan sheath around a single membrane, while gram-negative bacteria typically build a thin layer of peptidoglycan between 2 membranes. Water constantly enters bacterial cells by osmosis, building up pressure on the cell membrane. Peptidoglycan allows the cell to resist this pressure by providing structural support for the membrane. Penicillin prevents bacteria from building their peptidoglycan layer, causing the bacteria to burst under pressure. Let's take a closer look at how penicillin acts at the molecular level. Peptidoglycan is made up of small building blocks. each composed of 2 sugars connected to a short chain of amino acids with a peptide bridge extending to the side. These sugars are assembled into chains, which are then cross-linked via the peptide bridges to form a tough peptidoglycan matrix. The enzyme D-alanine carboxypeptidase transpeptidase, also known as penicillin binding protein, assists with peptidoglycan matrix assembly by creating the cross-links between the chains. Penicillin antibiotics block this enzyme by making a direct bond to a key serine amino acid in its active site. The active portion of penicillin is a beta-lactam ring. It is chemically reactive and opens up to form a bond to the active site serine. This inactivates the enzyme and prevents proper formation of the peptidoglycan matrix. The beta-lactam ring is found in both natural and synthetic antibiotics that are structurally similar to penicillin. Beta-lactam antibiotics are effective against many types of bacterial infections and have helped to save countless human lives. However, bacterial communities are living things, and like any other living thing, they adapt to survive. To protect themselves from antibiotics, bacteria developed resistance mechanisms. For example, MRSA, a strain of Staphylococcus aureus, expresses penicillin binding protein 2A, which has an altered active site that doesn't bind beta-lactam antibiotics. Bacteria can also express special beta-lactamase enzymes, which bind to beta-lactam antibiotics and break the essential beta-lactam ring, making the antibiotic ineffective. Medical scientists work quickly to develop new drugs to fight resistance. Some of these drugs block beta-lactamases directly, leaving the antibiotic free to inhibit penicillin-binding proteins. Other new beta-lactam drugs are either not recognized or not broken down by some beta-lactamases. Unfortunately, in the face of overuse and misuse of antibiotics, bacteria continue to develop ways to resist the action of new drugs. Many genes that are crucial in antibiotic resistance are encoded on small circular pieces of DNA called plasmids. These plasmids can be passed from one population of bacteria to another, and then from generation to generation. One such particularly dangerous gene is NDM1. Bacteria possessing this gene can build the New Delhi metallobeta-lactamase enzyme. These types of enzymes can break down almost all known beta-lactam drugs, posing a major global health threat. Today, less than 100 years since Fleming's discovery, we are in the middle of an arms race with pathogenic bacteria. Using our growing knowledge of the structural biology of bacterial enzymes, the medical community is looking for new ways to target the weak points of bacteria.
So just to recap, normal cell wall synthesis occurs when the bacterial enzyme transpeptidase makes peptide bonds between the amino acid strings in the peptidoglycan cell wall. When bacteria are sensitive to beta-lactam antibiotics, those antibiotics come into the cell, they bind transpeptidase and inactivate it. No peptide bonds are formed, and as the cell wall weakens, water rushes in and the cell dies. Bacteria have become resistant to beta-lactam antibiotics by 2 mechanisms. The one that we're going to focus most on is by the production of an enzyme called a beta-lactamase, which is often delivered to the cell via transformation or conjugation on a plasmid. So beta-lactamase, when it's present in the cell, makes the cell resistant because it is transcribed and translated into this protein, which binds the beta-lactam antibiotic and inactivates it. Once that's happened, transpeptidase is free to continue forming the peptide bonds that it always made. The cell wall maintains its structure, and the cell is resistant to the antibiotic. In order to combat that, combination drugs, which include clavulanic acid, in addition to the beta-lactamase, are used to make bacteria who have this beta-lactamase enzyme resensitized to the beta-lactam antibiotic. So the clavulanic acid binds the bacterial beta-lactamase. That allows the beta-lactam antibiotic to bind the bacterial transpeptidase. No peptide bonds are formed, the cell wall weakens, and the cell dies. So one major form of resistance to beta-lactam antibiotics is the inactivation of the beta-lactamase by cleavage of the beta-lactam ring, which we saw in the video. And the crazy thing is, this enzyme was present in bacteria before the antibiotic penicillin was ever approved. There's evidence in 1940, 3 years before penicillin was approved, of this penicillinase, this enzyme found in the population of bacteria. Because contrary to what that video sort of made it sound like, bacteria don't make these enzymes in response to the antibiotics. These enzymes are present in the population and they become useful in the presence of the antibiotic. It's just a matter of natural selection. Just like the long-necked giraffes are more likely to survive during a drought when they alone can reach the leaves at the tops of the trees, when an antibiotic is given to a host, then all of a sudden the bacteria who already have resistance to that antibiotic are the best surviving. Everybody else dies, they survive, and the antibiotic resistance spreads. Now, the problem is that these antibiotic resistances are becoming more and more common as we use antibiotics. So by 1979, something called an extended spectrum beta-lactamase was found. And this allowed bacteria to not only be resistant to penicillin, but resistant to cephalosporins. So that was combated by researchers by treatment with carbapenems, which are a later generation beta-lactam antibiotic. By the 1990s, carbapenemases were found in microbial populations, and these carbapenemases could kill the carbapenem antibiotic. So again, you had bacteria becoming resistant. And the problem with these carbapenemases that they found is that they're incredibly stable. We also have cephalosporinases, and as was mentioned in the video, that New Delhi metallobeta-lactamase is one of those incredibly stable, very resistant beta-lactamases, which does not have any problem with any of the antibiotics. And these are becoming more and more common. In the Petri dish there, you see bacteria growing. On the right-hand side, the E. coli that are shown are sensitive to the two beta-lactam antibiotics and don't grow in that zone of inhibition around the antibiotics. Unfortunately, the bacteria on the left are resistant to the beta-lactam antibiotics. The one at the top has a bit of inhibition, but still is considered resistant. And at the bottom, you can see the E. coli there has gained a resistance mechanism that has made him completely resistant to that antibiotic. As was mentioned in the video, there is a new beta-lactam antibiotic called New Delhi metallobeta-lactamase 1, which was initially isolated in India, which is where it gets its name, in 2009 in a Klebsiella pneumoniae. And by 2015, this New Delhi metallobeta-lactamase was found in bacterial isolates in 70 countries. By 2010, it was in England. It had found its way to the United States and Japan. It was in India. And frighteningly, there was a Canadian case where no travel to any of these areas had been noted. And by the time they published a paper on this novel beta-lactam antibiotic in 2010, there were 37 cases in the United Kingdom, 143 in India. It was plasmid-borne, and it was only sensitive to a highly toxic last resort antibiotic called colistin. And again, frighteningly, New Delhi metallobeta-lactamase has been found in drinking water and sewage in India. In 2012, it was found in a hospital-acquired infection, Acinetobacter. In 2013, the gene for the New Delhi metallobeta-lactamase 1 enzyme survived Chinese wastewater treatment. And a little bit of good news before we move on. In 2014, Aspergillus marasmine A from Aspergillus was shown to turn off the NDM1 beta-lactam antibiotic and restore susceptibility in rodents. So as you can see, this is a constant battle. The biological warfare between clinicians and researchers and microbial pathogens is constantly raging, and we're going to really see that occur on the next slide when we look at this biological warfare occurring not all over the planet, but within one single person in a 2009 case study. So in 2009, a 44-year-old woman who had undergone small bowel transplantation in 2005 for short gut syndrome as the result of a dissection of her mesenteric artery during a hysterectomy showed up with an infection. She was admitted to the hospital in 2008 with an admittance diagnosis of septicemia of mixed organisms, E. coli and Enterobacter cloacae. She was admitted. She's in the hospital by July of that year. She has a urinary tract infection of both E. coli and Proteus mirabilis. She's still in the hospital. The next month, she has an E. coli urinary tract infection, which is sensitive to imipenem, but is also co-infected with Klebsiella pneumoniae, which is imipenem resistant and ertapenem resistant. The next month, still in the hospital, she gets an abdominal infection of E. coli and Klebsiella pneumoniae, and guess what? They're both resistant to both antibiotics. So she's still in the hospital. November shows up. She's got a sputum that's positive for Pseudomonas aeruginosa, Serratia marcescens, and Klebsiella pneumoniae. Come December, she's got a multi-drug resistant pneumonia of Acinetobacter baumannii and Morganella morganii. And in January of 2009, her sputum is positive for Serratia marcescens, which is ertapenem and imipenem resistant. So what you're seeing in this woman is multiple infections occurring over time in the same person, and they're sharing antibiotic resistance with one another. And just to give you an idea, when we say that an organism is resistant to beta-lactam antibiotics, we're not talking about one or two antibiotics. We're talking about dozens of antibiotics. We're talking about resistant to narrow spectrum antibiotics like penicillin. We're talking about resistance to moderate spectrum antibiotics like amoxicillin or ampicillin, broad spectrum antibiotics like augmentin, which are that combination of amoxicillin and clavulanic acid, extended spectrum beta-lactams, cephalosporins, first, second, third, fourth generation, and then our biggest, baddest beta-lactam antibiotics, the carbapenems and penems, which have the broadest spectrum out there. Imipenem, which includes celastatin, which was that last resort antibiotic that's used against Nutelli beta-lactamase. And so saying that something's resistant to beta-lactam antibiotics is really to say that you are left with only last resort antibiotics in order to combat infection. So the obvious question is then, how do all these bacteria become resistant to antimicrobials? And like there were 4 targets of antimicrobial agents, there are several mechanisms of antimicrobial resistance, or AMR. Number 1, a microorganism can block entry of the antibiotic. So they can block entry. So whatever receptor used to bind the antibiotic has been modified, doesn't bind it anymore, and now they're resistant. They can inactivate the enzyme. They can cut that beta-lactam antibiotic because they carry with them, say, the beta-lactamase gene, which is transcribed and translated into an enzyme which cleaves the beta-lactam ring. So they can block entry, they can inactivate the antibiotic or drug once it gets into the cell. they can pump the antibiotic out of the cell so it gets in, but it's pumped out so quickly that it never hits its target. Or, as was mentioned in that video, you can have a target which will not bind the antibiotic. So you might remember from that video, there was a transpeptidase that isn't bound by penicillin. And because it's not bound by penicillin, it can continue to do its very important job of building the cell wall without risk of inhibition by the antibiotic. Antimicrobial resistance is a huge problem throughout the world, and it's one that research in the United States has been pouring money and time into to understand, because in the United States annually 2 million infections and 23,000 deaths can be attributed to antimicrobial resistant microorganisms. So the Centers for Disease Control have set up this system through which all different microorganisms can be categorized in order to understand the kind of threat they are in this fight against antimicrobial resistance. And all of the microorganisms that are flagged here are called either urgent or serious or concerning. And urgent are high consequence antibiotic resistant threats because they have a significant risk which has been identified across several criteria, and they may not be widespread, but they have the potential to become so, and so they are thought to require urgent public health attention. Not that you need to memorize any of these three that we're about to talk about, but I just want to give you an idea of an urgent, a serious, and a concerning antimicrobial resistance threat. So the urgent threat that I've chosen is Clostridium difficile, which is one of these big three, but this one causes 250,000 infections per year and 14,000 deaths, and has a medical cost per year in excess of $1 billion. And so though it doesn't cause that many deaths, maybe compared to the carbapenem-resistant Enterobacteriaceae or drug-resistant Neisseria gonorrhea, may not have as many infections or as many deaths, but it has a huge monetary cost. And if we can cut down those costs, those dollars can be used in all of the other arenas of fighting antimicrobial resistance. So C. difficile, or C. diff as he's called in the hospital, is our example of an urgent resistant microorganism. An example of a serious infection, which is a significant antibiotic resistant threat, but for varying reasons, either it's low infection rates or reasonable availability of therapeutic agents, they're not considered urgent, but they're on the watch list. And one example of that is drug-resistant Campylobacter, which causes 310 infections a year that are drug-resistant, 1.3 million total, with 13,000 hospitalizations and 120 deaths per year in the United States. As we move down to concerning infections are those for which the threat of antibiotic resistance is low, or there are multiple therapeutic options for resistant infections, though they still can cause severe illness, and they're on the radar just to be watched and monitored in case incidents come out so that they can respond quickly. And one example of that is clindamycin-resistant group B strep, which are the Streptococcus agalactiae, which can cause very serious infections in newborns, and even though they're not very common, that's such a serious thing that's what causes them to be on the radar as a concerning infection. There are about 7,600 drug-resistant group B strep infections per year, which cause 440 deaths. There were 27,000 severe group B strep infections in 2011, and as I mentioned, they are common in neonates. And if you have been pregnant, you will know that pregnant women are swabbed for group B strep prior to delivery to make sure that they are not harboring asymptomatically any group B strep in the vaginal area, which would then be passed to the newborn at birth. So the obvious question, once we've said, what is resistance, where is resistance, is why is resistance? And the basic gist is every time you use an antibiotic, you increase resistance to that antibiotic, right? So number one, there are a lot of germs always in those populations before antibiotics are given. There are drug-resistant bacteria in that population, right? They are not there as a response. They are there already. So when you take antibiotics, you kill the bacteria that are susceptible, and those that are resistant stick around, and now the drug-resistant bacteria are allowed to grow and take over. And then, of course, they can share that resistance with other bacteria through transformation, conjugation, or transduction naturally, and you get faster resistance spread through the population of bacteria. Some data that support that hypothesis are shown here. You have bacterial count shown in black, antibiotic resistance shown in red. So at the initiation of antibiotic therapy on day 2, antibiotic resistance present but very low. But as soon as you give the antibiotic, though the population of bacteria decrease exponentially, the relative abundance of resistance in that population increases, which is not necessarily a bad thing. Remember, it's a relative abundance, right? And when you're taking an antibiotic, you're assuming the person's immune system is going to be able to take care of some of the problem. So if you can just get the number of bacteria down to a level that the immune system can handle, then you will survive, because the immune system will come in and they will kill the resistant bacteria. The problem occurs when you don't take antibiotics wisely, when you don't steward the antibiotics, and then you end up with increased numbers of bacteria such that the immune system can't overtake them, and now you have a resident population that is resistant to antibiotics. So we need to talk about not the use of antibiotics, but the appropriate use of antibiotics, because they are absolutely appropriate. They are absolutely essential. It's just essential that we use them wisely. So inappropriate use of antibiotic would include the use of outdated, weakened, or inappropriate antibiotics. And just some data for you from a 2016 study, 25% of prescriptions that are given for antibiotics are given inappropriately, and 35% on top of that are potentially inappropriate, right? And we want to remember, antibiotic use equals antibiotic resistant. For every use of antibiotics, you have resistance to those antibiotics. And one of the places that that's a real problem is prophylactically in animal feed, because when you use antibiotics, animals tend to be bigger, they tend to be healthier, right? So they produce more milk or they produce more meat. Another example of inappropriate use is long-term low-dose antibiotics, aerosolized pulmonary antibiotics in hospitals, and the big one is on you, failure to follow prescribed treatment. Do not stop your antibiotics early, because when you do, you are left with more than your immune system can handle in resistant bugs. So let's say you get an antibiotic-resistant infection. How does that then spread through the population so that it's not just found in humans, but it's found in the groundwater or in animal population? So an example of how antibiotic resistance spreads when, for instance, a person gets antibiotics and then develops resistant bacteria in his gut, and if he stays at home and in the general community, he can spread that to other people. If he goes to a care facility or a hospital, he will spread those germs to people there. And so people in the community might contract it, people in the hospital might contract it. If animals receive antibiotics, they also develop resistant bacteria in their guts, and those antibiotics can sometimes persist in meats when they're not handled or cooked properly and can spread to human beings. They can also spread to human beings because they move through the gastrointestinal tract of the animals, end up in the water, or end up on the people who are taking care of the animals, and then move their way into the community. So where are we with antimicrobial resistance? And as of a couple of years ago, because the data take a while to put together, every year in the United States, antimicrobial resistant infections cost $20 billion in healthcare costs and $35 billion in lost productivity. And every time you use an antibiotic, you select for resistance. For instance, penicillin. Resistance identified in 1940, antibiotic produced in 1943, antibiotic resistance spread by 1965. Tetracycline introduced in 1950, resistance found in 1959. Erythromycin introduced in 1953, resistance found in 1968. Methicillin introduced in 1960, methicillin-resistant staphylococcus found in 1962. Gentamicin introduced in 1967 and resistance to gentamicin found in 1979. Vancomycin-resistant enterococcus and staphylococcus found in 1988 and 2002, just about a decade and a half after vancomycin was introduced. Imipenem was introduced in 1985, imipenem-resistant Enterobacteriaceae found in 1998. And the list goes on and on. Notice introduced antibiotic levofloxacin resistance on the same year. A year later, we have resistance in Staphylococcus, and a year later, we have resistance in Staphylococcus. So we need to find new antibiotics is the take-home message for that. But the problem is that in order to find a new antibiotic, on average, you're going to take 12 years and it's going to cost you more than $350 million. And what that means is over time from 1980 to the present, the number of antimicrobial drug applications has decreased year after year after year. And the reason for that is because it takes a long time to make sure that something is safe. FDA approval involves drug development, which takes about 2.5 years, and only about 1 in every 1,000 of those drugs that are developed will go to the FDA for investigative drug trials. If they do, they'll enter phase 1, which takes about a year and establishes the safety of the drug. If they survive that process, they move to phase 2, which takes about 2 years and assesses the effectivity of the drug or the antibiotic. Then it moves into phase 3, which is a 3-year process, which assesses the effectivity and adverse reactions. And then you have about 100,000-page application for approval, and 2.5 years and $200 million after that FDA testing is done. You can move to phase 4, which is post-marketing studies and monitoring. And this is a big business. $12.6 billion are invested per year by pharmaceuticals in order to try to find these antimicrobial agents. So the research has to be prioritized. And the research is prioritized based on how treatable, how deadly, what a healthcare burden it is in the environment, whether resistance is trending toward resistance, how prevalent resistance is, how easily transmitted the organism is, whether or not the organism is found in the community and also in healthcare environments, whether it can be prevented easily in healthcare settings, and whether it can be prevented in the community. So we're prioritizing antimicrobial research, and at the same time, we're fighting back against antimicrobial resistance. And we're fighting back on 4 fronts. Number 1, we're trying to prevent infections in order to prevent the spread of disease. Number 2, we're tracking incidence of antimicrobial resistant infections. Number 3, we're pushing to improve antibiotic prescription and stewardship. And number 4, we're developing new drugs and diagnostic tests in order to fight back against antimicrobial resistance. And we fight back because more than 50% of antibiotic prescriptions are not needed and are not optimally effective, and all antibiotic use causes C. difficile infection. And all drugs, no matter how safe you think they are, have side effects, have possible allergic reactions, and they account for 140,000 emergency department visits per year. And in The pediatric population, children under 18, 1 out of every 5 emergency department visits for adverse drug reactions are due to antibiotic use. So if you don't need it, you shouldn't take it, not only because it increases antimicrobial resistance, but because there's no point in you increasing your likelihood of adverse effect. So one of the ways we can fight back against antimicrobial resistance is by diagnosing resistance and using antibiotics effectively. And one of the ways that we use antibiotics effectively is by understanding how much antibiotic a patient might need. And we do that by determining something called the minimum inhibitory concentration, the minimum concentration of an antibiotic required to inhibit the growth of a microorganism. And that is done clinically doing something called the epistolometer test, which is shown here. It's done on a rather large petri dish that is filled with blood agar. The organism is seated across the surface of the plate, which you can see here in white, and antibiotic-containing strips, which have a concentration gradient. They are most concentrated toward the middle of the plate and less concentrated as they move out. So those strips are placed on top of the seated agar, and they are allowed to grow, and you can see this red zone of inhibition where the blood agar is visible, where no microorganism has grown, and the minimum inhibitory concentration ends up being at the very bottom of that zone of inhibition. So the minimum inhibitory concentration of this TM antibiotic is 0.064. The antibiotic on the side of that has a minimum inhibitory concentration of about 0.064 as well, and then the minimum inhibitory concentration of the third antibiotic is 0.25. So this allows you to determine the therapeutic dose of an antibiotic so that you are inhibiting the infection and allowing the body's immune system to take part in the defense against the infection and clear out all of the underlying resistant microorganisms. Another useful analysis is knowing the minimum bactericidal concentration, which can't be seen with the epistolometer test because the microorganisms are grown in the presence of the antibiotic. We never know whether they're being inhibited or whether they've been killed. So in order to know the bactericidal concentration, you have to do something called the use dilution test, which involves exposing microorganisms to an antimicrobial agent and then removing the antimicrobial agent and testing to see whether they're still alive. So it begins with a series of test tubes which have an increasing concentration of antibiotic in the cultures, with low antibiotic concentration beginning on the left-hand side and ever increasing over to the right-hand side where you have the highest concentration of antibiotic. To each of those 6 tubes, you place the bacteria in question and you put them in the incubator to let them grow. At the lowest concentrations, the first 2 tubes, you see growth of the microorganisms with a minimum inhibitory concentration shown there in tube A, and then no growth in the remaining 3 tubes. The question is, in that group A, are the microorganisms inhibited from growing? Is it bacteriostatic or is it bactericidal? And in order to test that, you take all of the tubes in which the microorganism did not grow, as well as the dilution just above that, and you plate them with no antimicrobial agent. You put those in the incubator and you look for growth. So the tube where the bacterium was not inhibited serves as a positive control for the experiment. So you should see growth there. In tube A, which had the minimum inhibitory concentration, you see growth occurring in the plate. So though it was inhibitory at that concentration, it was not bactericidal. In addition to that, the next higher concentration was also not bactericidal, even though it was inhibitory. But then finally with tube B, you have a concentration that is both inhibitory and bactericidal. So this minimum bactericidal concentration is helpful when you have a rather serious infection or an infection in a person who is immunocompromised and you really can't expect the patient to take part in ridding themselves of the microorganism. So both have their place, and it really depends what's the microbial agent, what's the status of the patient, and multiple other things that would lend you information to decide whether you should go with a minimum inhibitory concentration or a minimum bactericidal concentration. So that's one way that we can fight back against antimicrobial resistance, is to diagnose resistance and use those antibiotics appropriately, right? Do away with this 50% of all prescriptions being unnecessary or not optimally effective, and really use those antibiotics effectively. We can also track infections, and that involves not just tracking an infection in a person, but tracking infections statewide, nationwide, and even internationally. And one really interesting study that was done that's helped to curb antimicrobial resistance is the rapid detection of carbapenem-resistant enterics, which you might remember, carbapenems are those sort of last resort beta-lactam antibiotics. And there was a 4-step process in place when these carbapenem-resistant enterics were identified in healthcare settings, and they included on-site infection control, screening of exposed contacts, so whether or not they were showing symptoms, and a facility-wide response with intervention until the transmission was controlled. And when they did that with these carbapenem-resistant enterics, they saw a drastic reduction in the percentage of these infections that were resistant to carbapenems. So the logical step was to apply that broadly, and when they did that in 2017, to extended spectrum beta-lactamase containing bacteria, they took this curve that had been moving along at about the same rate of infection over time and are attempting to duplicate in the extended spectrum beta-lactamase containing microorganisms what they have found to be successful with the carbapenem-resistant enterics. So we're diagnosing resistance, we're tracking infections in order to keep those resistant microorganisms sequestered, keep them from sharing that information with other microorganisms. And that's done typically by the Centers for Disease Control, who has regional labs across the United States, in the West, in the central United States, in the Midwest, up in the Northeast, Mid-Atlantic, and in the Southeast. And so that allows there to be these locations that are much easier to get to than the Centers for Disease Control headquarters in Atlanta, so that clinicians could get samples to these labs, diagnose and track these infections, and again, all of that with the aim of fighting back against these antimicrobial resistances. So just kind of a case study in fighting back by using antibiotics appropriately. You'll remember that penicillin was approved in the early 1940s to treat syphilis. It was also approved to use as a treatment for gonorrhea. By 1955, the dosage required to control gonorrheal infections had increased by 10 times. And by the 1960s, tetracyclines had replaced penicillin as the antibiotic of choice. In the 1980s, ciprofloxacin replaced the tetracyclines as the antibiotic of choice. And in 2004, cephalosporins were recommended by the World Health Organization to treat these gonorrheal infections, which are a worldwide problem. And today, the go-to treatment is really what would be considered a last resort treatment, and that is an injectable ceftriaxone in a single dose oral erythromycin. And that's done because there oftentimes are co-infection of people who have gonorrhea with chlamydia, and this ceftriaxone and azithromycin will treat both of those infections. It also confirms that the patient will adhere to the antibiotic therapy prescribed by the physician, because you give them the injection in the office and you watch them take the single dose oral azithromycin, and you know that they have been treated effectively. Well, the problem with that is that in 2013, a major study found that all treated gonorrheal infections had a 70% susceptible rate to ciprofloxacin and an 80% susceptible rate to penicillin. So the question is, are you using these broader spectrum antibiotics effectively and appropriately if you could go to something more narrow spectrum like penicillin? And the pushback on that is what I just mentioned, that giving them this last resort treatment confirms for the clinician they are being treated effectively, and it also is going to treat chlamydia, which is oftentimes comorbid with gonorrhea. So the thing to try to push for here is not necessarily to mandate the treatment, but to provide rapid diagnostics which will show whether or not these organisms in the clinic are susceptible to these more narrow spectrum drugs before the patient leaves the lab. And it's thought that that would reduce the use of this more broad spectrum ceftriaxone by about 66%. So that would be a great way to use narrow spectrum antibiotics when possible and save those last resort more broad spectrum antibiotics for use when they are necessary. So speaking of rapid diagnostics, we have a couple of different kinds of rapid diagnostics. We have nucleic acid amplification tests, so a PCR that actually looks for the genes for antibiotic resistance. And there is an sputum test, which is done by looking for tuberculosis that is resistant to rifampicin and things like that, and it within about 2 hours will tell you whether or not those antibiotics are useful against it, and that as compared to at least a 48-hour turnaround time for routine culture and sensitivity. Mass spectrometry is used to look for pathogens in blood and identify them and test them for sensitivity within about 8 hours. Something called PNA FISH, which can give you an identification and antibiotic sensitivity within 7 hours. And not to get ourselves wrapped up in all of the specifics of these, but these rapid diagnostic tests are really important, not just for identifying the microorganisms, which is a place where they've been utilized for years, but to look for antibiotic sensitivity in that same way. So not only to diagnose Streptococcus pyogenes in the doctor's office within 5 minutes, but to also know that that Streptococcus pyogenes is going to be sensitive to a narrow spectrum antibiotic before you leave the doctor's office. And again, that's all compared to routine culture and sensitivity testing in the lab, which takes at least 48 hours. Another really interesting way that clinicians are fighting back is through the use of alternate therapies. We talked in chapter 7 about controlling microbial growth by the use of bacteriophages, so inoculating intentionally with bacteriophage, which attack their bacterial pathogens and cure them. There are also some really interesting blood filters that work almost like dialysis, where the blood is filtered through something like this Seraph 100 blood filter, which binds pathogens, reduce the number of microorganisms in the blood by 85% in a single pass through this filter. And there are lots of different filters that are used to bind different kinds of pathogens, different kind of pathogenic toxins. So this is one way that physical removal, almost like a filtration of the microorganisms, is used to fight back, to do something besides giving a patient antimicrobial agents like drugs or antibiotics in order to cure them of infection. And finally, vaccines as a way of fighting back against antimicrobial resistance. As you can see from the top of this diagram, antibiotics, as soon as they're used, and we've heard this before through this chapter, use creates resistance. But vaccines have been formulated against dozens of antimicrobial agents, and smallpox has been eradicated, polio is near eradication, and none of them are perfect alternatives, but they're part of the arsenal that can be used along with the appropriate stewardship of antibiotics in order to fight antimicrobial resistance and allow us to keep that arsenal of antibiotics that we have for as long as possible. So again, antimicrobial resistance, ways that we're looking at to try to stop that is appropriate use of antibiotics, tracking of these resistant infections, stewardship of the antibiotics, and utilizing rapid diagnostics and alternate therapies, including vaccines, in order to stop the spread of antimicrobial resistance.